Welcome to another deep dive where we're going to be tackling some complex research. Today we're focusing on a study about a neurological disease in small Swiss hounds.
It's called neuronal ceroid lipofuscinosis, NCL for short. And this research is really unearthing some interesting genetic discoveries about it.
Yeah, the title, Intragenic Duplication Disrupting the Reading Frame of MFSD8 in Small Swiss Hounds with Neuronal Ceroid Lipofuscinosis, is a mouthful. We'll break it down, figure out what makes this so important. Let's start with the basics. What exactly is NCL, and why is it something we should be concerned about in these dogs?
So NCL is a group of lysosomal storage diseases. What that means is there's basically a malfunction in the cellular system that's responsible for breaking down waste products. This leads to a buildup of certain materials inside the cells, and ultimately it causes progressive neurodegeneration.
So it affects their brains and nervous systems. The symptoms probably reflect that damage, huh?
Absolutely. This particular study looked at two small Swiss Hound littermates who, around 12 months of age, started showing signs of progressive ataxia. They also had cognitive decline and vision loss.
Oh, that sounds really tough, especially for such young dogs.
It is. NCL is progressive and severe. Unfortunately, both of these dogs had to be euthanized.
The study mentions a pathological exam. They did a pretty detailed one on one of the dogs, right? What did they find that sort of solidified the NCL diagnosis?
The exam showed both cerebral and cerebellar atrophy, basically shrinkage in certain areas of the brain, and they also saw an accumulation of what's called autofluorescent material inside the neurons. Both of those are classic signs of NCL.
So how did they figure out what was causing NCL in these specific dogs?
They used whole genome sequencing on one of the dogs, which let them analyze its entire genetic code.
Right.
What they found was a pretty sizable duplication in the MFSD8 gene on chromosome 19. It was 18,819 base pairs long.
Okay, so there's this duplication in the MFSD8 gene. What does it mean for how the gene actually works?
Think of a gene like a set of instructions for building a protein. When you have a duplication, it's like copying and pasting a chunk of those instructions. That messes up the entire reading frame of the gene, so the protein production is disrupted.
And is this MFSD8 gene important for normal brain and nervous system function?
Definitely. MFSD8 is key for lysosomal function. Remember, those are the cellular recycling centers. When it's disrupted, those lysosomes can't do their job. We start to see that buildup of waste material, and that's what ultimately leads to those neurological symptoms.
So both logs had this duplication in the MFSD8 gene. Did they inherit it? And if so, how?
That's the crux of the matter. Both dogs had the duplication in a homozygous state, meaning they got a copy of the mutated gene from both parents. They did a pedigree analysis of 106 small Swiss hounds, and it confirmed this NCL is inherited in an autosomal recessive pattern.
Can you explain what autosomal recessive inheritance means, especially when it comes to breeding these dogs?
Sure. Autosomal simply means that the gene is on one of those non-sex chromosomes. And recessive means that an individual needs two copies of the mutated gene, one from each parent, to actually develop the disease. If a dog inherits just one copy, they become a carrier, but they won't show any symptoms.
So that means genetic testing is super important to breeders so they don't pair two carriers without realizing it.
Exactly. If breeders can identify those carriers, they can make informed choices about breeding and prevent producing puppies that will be affected by NCL.
This MFSD8 gene, is it connected to NCL in any other species?
It is. Mutations in MFSD8 actually cause a specific type of NCL called NCL7, and we see it in humans and other mammals as well.
So what we're learning about small Swiss hounds might actually have implications for understanding NCL in other species.
Precisely. The findings from this study add a valuable piece to the puzzle when it comes to understanding the broader genetic basis of NCL as a whole.
This is really highlighting how much we can learn from studying canine genetics, isn't it? And how it can potentially help us understand these kinds of diseases even in other species.
It really does. It emphasizes just how much we still have to discover and underscores the importance of this research so we can improve the health of animals and humans alike.
Now, the study gets into some specifics about this duplication in the MFSD8 gene. Can you walk us through what they found?
They were able to identify the exact locations of the breakpoints. That's where the genetic material is repeated. They found it in intron 3 and exon 12 of the gene.
Why is knowing the exact location of those breakpoints so important?
It helps us understand how that duplication is actually messing with the gene's reading frame. Remember, exons are those coding regions of the gene, the parts that actually carry the instructions for building the protein. And introns are those non-coding regions that are kind of interspersed between the exons.
So when you get this duplication, it basically scrambles the order of the exons and the introns.
You got it. It's like if you inserted a random sentence right in the middle of a paragraph, suddenly it doesn't make sense anymore. This disruption in the reading frame makes it impossible for the cell to properly read the genetic instructions, so you don't get a functional protein.
The study also talks about how they actually confirmed this duplication. They used PCR, didn't they?
Yes, they used polymerase chain reaction, or PCR. It's a technique that helps amplify a specific fragment of DNA. In this case, they targeted a fragment that spanned the junction point of the duplicated sequences. This helped them visualize the duplication, which wouldn't be there in a dog without the mutation.
And they went even further to quantify the duplication using something called droplet digital PCR, or
The ddPCR is super sensitive. It allowed them to actually count the number of copies of the duplicated region. They found that the affected dogs had 4 copies, carriers had 3, and dogs without the mutation had 2.
This is a really detailed level of analysis, isn't it? It really highlights just how complex these genetic analyses are and how rigorous these methods need to be.
Absolutely. It demonstrates just how much information we can get from these modern genetic techniques and how it all contributes to a deeper understanding of these diseases.
So far, we've talked a lot about how this duplication affects the MFSD8 gene. But what about the protein itself? What does it actually do in the body?
The MFSD8 protein acts as a chloride channel. What that means is it regulates the flow of chloride ions across the lysosomal membrane. This is crucial for maintaining a proper balance of ions inside the lysosome.
So chloride ions are really important for the lysosome to do its job properly. How does that work?
Lysosomes need a very specific environment internally to effectively break down those waste products. If the pH or the membrane potential is off, they can't function correctly.
So by regulating that chloride flow, MFSD8 is basically helping to keep that lysosomal environment in check.
Precisely. When MFSD isn't working properly, like we see in these dogs, that balance gets disrupted, and you get the accumulation of waste products, which then leads to the onset of NCL.
It's amazing how all of these pieces, genetics, cellular function, and disease, are so intricately connected, isn't it?
It really is, and it underscores why this kind of research is so important. The more we understand these connections, the closer we get to developing effective therapies for these diseases.
The researchers used what are called the ACMGM guidelines to figure out how likely it is that this MFSD8 duplication is actually causing the disease. Can you walk us through those guidelines again?
Sure. They're basically a set of criteria that are used to figure out how strong the evidence is linking a genetic variant to a certain condition. They're really important in human and veterinary genetics because they help standardize how we interpret genetic variants and make sure we're classifying them accurately.
What were the specific criteria they looked at when they were evaluating this duplication?
There were 3 main criteria. First, they determined that this duplication was likely a null variant, meaning it basically completely shuts down the function of the gene. They based that on how the duplication disrupts that reading frame that we talked about earlier.
Right, because that jumbled reading frame is probably preventing the cell from producing any functional protein at all.
Exactly. The second criterion was co-segregation, which basically means that they were looking to see if everyone who has NCL also has this duplication, and if everyone who is healthy lacks it.
And they found a pretty clear correlation there, right? Supporting the idea that this variant is actually causing the disease.
Right. And then the final criterion they looked at was the specificity of the clinical signs. So they were looking at whether the symptoms in these dogs really pointed to NCL specifically, and were there any other potential causes that could explain those symptoms.
And those clinical signs, the progressive neurodegeneration and the accumulation of that autofluorescent material, those are really hallmarks of NCL, aren't they?
Exactly. There weren't any other obvious explanations for the symptoms these dogs were showing. So based on all 3 of those criteria, they felt pretty confident in classifying this MFSD8 duplication as pathogenic.
They also looked at a control group of small Swiss hounds as part of their analysis, right? To help strengthen their findings.
Yes. They look at 104 small Swiss hounds, and none of them had the duplication in a homozygous state, which further supports their conclusion.
So all of the evidence is really pointing to this MFSD8 duplication as being the culprit for NCL7 in these dogs.
It really is. It's a great example of how genetics can be used to really untangle these complex diseases. And now that we've identified this genetic cause, the next step is figuring out how we can use this information to help these dogs.
That's the burning question, isn't it? What are the practical implications of this discovery for small Swiss hounds?
The most immediate impact is that it opens up the possibility of developing a genetic test for this specific mutation.
That could be a real game changer for breeders, couldn't it? If they're able to screen for carriers and make informed decisions about breeding, it could really help to reduce the number of puppies that are born with this disease.
Absolutely. It's a powerful example of how understanding genetics can directly translate into better health and well-being for these dogs. And it goes beyond small Swiss hounds, too.
What do you mean?
Well, remember we talked about how MFSD8 mutations also cause NCL7 in humans? So what we're learning from this study in dogs could actually provide valuable insights that could help with research and maybe even lead to treatment strategies for human NCL7 as well.
So it's not just about one breed, it's about advancing our understanding of a disease that affects multiple species.
That's exactly right. That's what's so exciting about this kind of research. The ripple effects are much broader than just the initial focus of the study.
Okay, so we now know that this specific duplication in the MFSD8 gene is causing NCL7 in these small Swiss hounds. Where does the researchers go from here? What's the next step?
That's a great question. One really promising avenue is to explore potential therapies for NCL7.
What kind of therapies are we talking about?
Well, one really exciting possibility is gene therapy.
And what is gene therapy exactly?
Basically, it involves delivering a functional copy of the MFSD8 gene directly to the affected cells. It's cutting-edge stuff. And it holds a lot of promise for treating a whole range of genetic disorders.
So the idea is to basically correct that underlying genetic defect. That sounds incredibly complex.
It is challenging, but researchers are making some really significant progress in this field. And studies like this one provide a strong foundation for future research into gene therapy for NCL7, research that could potentially benefit both dogs and humans.
It's remarkable how one study can open up so many possibilities for the future, isn't it? It feels like we're on the cusp of some really groundbreaking advancements in veterinary medicine.
We are, and this is just one example of how canine genetics is helping us unlock the secrets of these complex diseases and paving the way for more effective diagnoses and treatments for animals and people.
We've covered a lot of ground already, but I want to go a bit deeper into some of the finer points of this study. The article has a bunch of figures and tables that show their findings visually. Can you walk us through some of those?
Absolutely. Let's start with Figure 1. This figure shows a pedigree of the small Swiss Hound family that was part of the study. It really clearly illustrates that autosomal recessive inheritance pattern of the MFSD8 duplication that we were talking about earlier. You can see how both of the affected dogs inherited a copy of the mutation from each parent.
And you can see in the pedigree that the parents and one unaffected sibling are carriers, meaning they have one copy of the mutation and one copy of the normal gene.
Exactly. Now let's move on to Figure 2. This figure shows the results of that whole genome sequencing analysis, and it highlights the specific region on chromosome 19 where the MFSD8 duplication is located.
It's pretty amazing to see how they were able to pinpoint the exact location of that duplication within the entire genome of the dog, isn't it?
It really speaks to the power of these sequencing technologies. And Figure 3 shows the actual structure of the MFSD8 gene itself and the location of the duplication. You can see how the duplication is disrupting that reading frame of the gene.
This figure really helps visualize how that scrambled reading frame would prevent the production of a functional protein.
Right. Now let's take a look at Table 1. This table summarizes the clinical signs that were observed in those two affected dogs. You can really see how the neurological decline was progressive. The symptoms started around 12 months of age and got worse over time.
It's heartbreaking to see how quickly their health deteriorated.
It really is. It highlights just how devastating this disease can be.
Now, let's look at Table 2. This table shows the results of the genetic testing that they did for the MFSD8 duplication.
You can see that both of the affected dogs were homozygous for the duplication, meaning they had those two copies of the mutation. And the parents and the one unaffected sibling were heterozygous, meaning they had one copy of the mutation and one copy of the normal gene.
And it's important to note that none of the dogs in that control group were homozygous for the duplication, which again really supports the idea that this particular variant is what's causing the disease in small Swiss hounds.
Exactly. All of these figures and tables really help to bring the research to life, don't they? They make the findings more accessible, and they really highlight the rigorous methods that were used in the study.
They definitely do. They provide a visual representation of all of that data, and they really help to drive home the link between that MFSD8 duplication and NCL7 in small Swiss hounds.
It's a great example of how genetics can be used to help us understand and potentially address these complex diseases.
This has been incredibly insightful so far. But there's still so much more to explore. We've just scratched the surface of this research.
And that's what makes this field so exciting. There's always something new to uncover, always another layer to peel back.
So join us for part 2, where we'll continue our deep dive into this groundbreaking research on NCL7 and Small Swiss Hounds.
We'll delve even deeper into the implications of these findings. And we'll explore the potential for future therapies that could benefit not only these dogs, but potentially humans as well.
And we'll continue to break down the science in a way that's both accessible and engaging.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging. Welcome back to our deep dive on NCL7 in Small Swiss Hounds. In part 1, we talked about how a duplication in the MFSD8 gene messes up its function, and how that leads to this neurodegenerative disease. Now, let's get into the nitty-gritty of how the researchers did this study.
You mentioned they use linkage analysis and autozygosity mapping. Can you break down what those are and how they used them here?
Linkage analysis basically looks at how traits are inherited across generations. It helps map where genes are located on chromosomes. If genes are often inherited together, it suggests they're close to each other on a chromosome.
So it's about figuring out which genes are neighbors.
Exactly. Now, autozygosity mapping is different. It's about finding regions in an individual's genome where they've inherited identical gene copies from both parents. So they're homozygous for that region.
So linkage analysis looks across generations. Autozygosity mapping zooms in on one individual's genome.
That's a good way to put it. The researchers used both of these techniques to try and pinpoint the gene responsible for NCL7 in these dogs.
How did they use those methods to zero in on the MFSD8 gene?
First, they did linkage analysis using something called single nucleotide polymorphism. It's SNP genotype data from the affected dogs, their unaffected sibling, and their parents. They found 12 genomic intervals that were likely to contain the gene they were looking for.
So they narrowed it down to 12 suspect areas.
Yes. Then they did autozygosity mapping using SNP genotype data from the 2 affected dogs. This showed several homozygous regions shared by both dogs, suggesting the gene was hiding in one of those regions.
Okay, so they combined the linkage analysis, which pointed to those 12 candidate regions, with the autozygosity mapping, which highlighted those shared homozygous regions. That helped them really refine their search, huh?
Exactly. One homozygous region they found through autozygosity mapping actually overlapped with one of the intervals from the linkage analysis. That overlapping region on chromosome 19 contained the MFSD8 gene.
So using both techniques together helped them say, aha, MFSD8 is our likely culprit.
Exactly. It's a good example of how powerful it can be to combine different genetic techniques, especially when you're trying to understand these complicated diseases.
They didn't stop at just identifying the gene, right? They went on to confirm the duplication and really analyze what it was doing.
Right. They used several techniques to understand the specifics of this mutation. One was PCR, polymerase chain reaction, to amplify a DNA fragment. That fragment spanned the junction point of that duplicated sequence.
So basically highlighting the exact spot where the genetic material repeats.
Yes. And this PCR analysis confirmed that the duplication was present in both of those affected dogs.
And they didn't just confirm it. They used that droplet digital PCR, the ddPCR, to quantify it, right? To really measure it.
You got it. ddPCR is really sensitive, which allows researchers to count the number of copies of specific DNA sequences. What they found was that the affected dogs had 4 copies of that duplicated region. The carriers had 3, and those without the mutation had 2.
That's an incredible level of precision. Now, to really get into how this duplication affects the gene's function, let's talk about the structure of the MFSD8 gene itself.
Sure. Remember, a gene is essentially a set of instructions for building a protein. It's written in the language of DNA, and DNA is made up of those 4 building blocks, the nucleotides.
Adenine, thymine, guanine, and cytosine, or A, T, G, and C, right?
You got it. The order of those nucleotides in the DNA sequence of the gene tells the cell the order of amino acids in the protein.
And amino acids are the building blocks of proteins. Their specific sequence determines the protein's structure and ultimately its function.
Exactly. So if you change the DNA sequence, you're changing the protein structure and function. And that's what this duplication in the MFSD8 gene is doing. It's disrupting what's called the gene's reading frame.
What do you mean by reading frame?
Imagine the DNA sequence like a sentence where the nucleotides are the letters. The cell reads this sentence in groups of 3 nucleotides. Those are called codons. Each codon specifies a particular amino acid.
So it's like a secret code the cell has to crack. reading 3 letters at a time.
A good analogy. Now, the reading frame refers to where those 3-letter codons start. If you shift the reading frame, even by 1 nucleotide, you end up reading the wrong codons. And that leads to the production of a completely different protein.
So it's like if you started reading a sentence from the wrong word, it's not going to make sense.
Exactly. And that's what's happening with this MFSD8 duplication. It's shifting that reading frame, which means the cell can't produce a functional MFSD8 protein. And that throws off the lysosome function, leading to NCL7 in these dogs.
It's amazing how a small change in the DNA sequence can have such a huge impact.
It is, and it highlights why understanding the genetic basis of these diseases is so crucial.
You mentioned earlier that MFSD8 mutations are also linked to NCL7 in other species, even humans. Can you talk a bit more about that?
Well, the link between MFSD8 and NCL7 was actually first discovered in humans. Mutations in this gene can cause a rare type of NCL called variant late infantile NCL. It usually shows up between 2 and 6 years of age.
So the same gene that's causing NCL7 in these small Swiss hounds is also involved in a similar disease in people. That really reinforces the idea that studying these dogs can teach us a lot about the human form of the disease, doesn't it?
Absolutely. This study really highlights the value of these canine models for NCL research. What we learn from dogs could potentially lead to treatments that could help both humans and animals that are affected by these conditions.
It's a great reminder that human and animal health are really interconnected. Now let's focus a bit more on the MFD8 protein itself. You mentioned it acts as a chloride channel. Can you remind us what that means and why it's so important?
Sure. A chloride channel basically acts as a gatekeeper in the cell membrane. It controls the flow of chloride ions in and out of the cell, which is super important for all sorts of cellular functions.
So MFSD8 is like a bouncer for chloride ions.
That's a good way to think about it. And specifically, the MFSD8 chloride channel is located in the lysosome membrane. We've talked about lysosomes being those cellular recycling centers. They need a very specific environment inside them to function properly. That includes having the right balance of ions.
So MFSD8 is helping to maintain that balance within the lysosome by regulating the flow of chloride ions.
Exactly. When MFSD8 isn't working properly, like in these dogs with the duplication, that balance gets messed up. Waste products start to accumulate inside the lysosomes, and that's when we see the development of NCL.
It's like a chain reaction. The mutation disrupts the chloride transport, the lysosome's environment gets thrown off, waste builds up, and eventually you get those neurological symptoms of NCL. It's fascinating how it all works together, but also pretty complex.
It is. It highlights how intertwined genetics, cellular function, and disease really are.
The researchers use the ACMGMP guidelines to evaluate how likely it is that this MFSD8 duplication is actually causing the disease. Can you walk us through those guidelines again?
Those guidelines basically provide a framework for evaluating the evidence that links a particular genetic variant to a specific condition. It helps standardize how we interpret those variants, and it helps us to make sure that we're classifying them correctly.
What were the specific criteria that they looked at in this case?
They considered 3 main things. First, they looked at whether the variant is what we call a null variant, meaning it completely wipes out the function of the gene. They concluded that this MFSD8 duplication likely fits that description because of how it disrupts the reading frame of the gene.
Right, because that jumbled reading frame probably prevents the cell from making any functional protein at all.
Exactly. The second criterion was co-segregation. Basically, they were looking to see if everyone who has NCL also has this specific genetic variant, and if everyone who doesn't have NCL lacks it.
And they found a pretty strong correlation there, right? Which supports the idea that this variant is what's causing the disease.
That's right. And the final criterion they looked at was the specificity of the clinical signs. They were asking, do the symptoms that we're seeing in these dogs really point to NCL? And are there any other possible explanations for those symptoms?
And those clinical signs, the progressive neurodegeneration and the accumulation of that autofluorescent material, those are the hallmarks of NCL, aren't they?
Exactly. There weren't any other obvious explanations for what these dogs were experiencing. So based on all 3 of those criteria, they felt pretty confident in saying that this MFSD8 duplication is pathogenic.
And they also looked at that control group of 104 small Swiss hounds. And none of those dogs had the duplication in a homozygous state, which further strengthens their conclusion.
Right. So all of that evidence really points to the MFSD8 duplication as the root cause of NCL7 in these particular dogs.
Now, beyond the science itself, what are the real-world implications of this discovery, especially for small Swiss hounds?
The most immediate impact is that it paves the way for developing a genetic test specifically for this mutation.
That's huge for breeders, isn't it?
Yeah.
It means they can screen their dogs for this mutation, identify carriers, and make informed breeding decisions. And ultimately, hopefully reduce the number of puppies born with NCL.
Absolutely. It's a powerful example of how understanding genetics can have a direct impact on improving the health and well-being of a breed, but it also goes beyond just small Swiss hounds.
In what way?
Well, remember we talked about how MFSD8 mutations can cause NCL7 in humans too? The findings from this study in dogs could have broader implications for research and potentially even treatment strategies for human NCL7.
So it's not just about one breed of dog. It's about expanding our understanding of a disease that affects multiple species.
That's the beauty of this kind of research. It has these ripple effects that reach far beyond the initial focus of the study.
This has been incredibly informative. We've gone through the science behind this discovery, the methods they used, and we've even started talking about the implications for both dogs and humans. What else can we learn from this research?
One thing we haven't really touched on is the importance of genetic diversity. This study focused on a specific mutation in small Swiss hounds, but it's important to remember that genetic diversity is crucial for the overall health and resilience of any population.
What do you mean by genetic diversity, and why is it so important?
Think of it as a buffer against disease and environmental changes. A diverse gene pool makes it more likely that at least some individuals will have the traits that help them survive and thrive even when things change.
So it's like an insurance policy for a population.
Exactly. In dog breeds, when you have selective breeding for certain desirable traits, sometimes you inadvertently reduce genetic diversity, and that can make certain genetic diseases more common.
So even though we have tools like genetic testing and selective breeding to manage these diseases, it's important to make sure we're also preserving genetic diversity.
Absolutely. It's a balancing act. It requires careful planning and collaboration between breeders, geneticists, and veterinarians.
That makes sense. You mentioned that genetic testing is key for identifying carriers of this MFSD8 duplication in small Swiss hounds. What does that look like in practice for breeders?
Genetic testing for dogs is much easier and more affordable than it used to be. There are a lot of companies out there that offer these tests for different breeds.
So how does a breeder actually get their dogs tested?
It's usually a pretty simple process. They can order a test kit online or through their vet. The kit usually includes swabs for collecting DNA from the dog's cheek.
And then they just send those samples back to the company.
Yep. The company analyzes the dog's DNA and looks for specific genetic variants, including that MFSD8 duplication in this case, and then they send the results back to the breeder.
So breeders can use those results to make informed breeding decisions and avoid pairing two carriers together.
Exactly. It's a really powerful tool for promoting responsible breeding practices.
We've also talked about gene therapy as a potential treatment for NCL7. Can you expand on what that is and how it might work in this case?
Gene therapy is all about trying to alter genes to treat or prevent disease. It's still relatively new, but it has huge potential for tackling a wide range of genetic disorders, including NCL.
How would it work for NCL7 specifically?
The idea is to deliver a working copy of that MFSD8 gene directly to the cells that are affected by the disease. They often use modified viruses called viral vectors to do this.
So they're using viruses to deliver good genes instead of bad ones.
Yep, it's about taking advantage of the way viruses naturally infect cells. But instead of causing harm, we're using them to deliver therapeutic genes.
So if you could deliver a healthy MFSD8 gene to a dog with NCL7, it could potentially correct that underlying genetic defect and stop the disease from progressing.
That's the hope. Gene therapy is still in its early stages, and there are a lot of challenges that need to be overcome before it can be used to treat NCL7. But research like this study lays the foundation for those future advancements.
It's exciting to think about the potential that gene therapy has, not just for dogs, but for humans as well. This deep dive into canine genetics has been so eye-opening. We've explored NCL, talked about the role of the MFSD8 gene, learned about this specific genetic duplication, and even touched on future therapies like gene therapy. It really highlights the impact that scientific research can have on both animals and humans.
It does. It emphasizes how connected we all are and how important it is to keep pushing forward with this research.
We've covered a lot of ground in this second part of our deep dive, but there's still more to discuss. There are more fascinating details and implications to uncover.
So join us for the third and final part. We'll explore the significance of these findings in even more depth, and we'll talk about how this research could shape the future of veterinary medicine.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging. And we're back for the final part of our deep dive into this NCL7 research in small Swiss hounds. We've learned a lot about the genetics of the disease, we've talked about the methods, and we've even touched on some potential therapies. Now let's step back and think about the big picture. What does this all mean moving forward?
You know, we've spent a lot of time talking about MFSD8 and its role in NCL7, but I think it's important to zoom out a little and think about the broader implications of this kind of genetic research.
You're right. This study is just one example of how understanding canine genetics can have these really wide-reaching effects.
Exactly. When we study dogs, we're not just helping them, we're also learning things that can help us understand human diseases, and maybe even treat them. It really highlights that One Health concept, the idea that human, animal, and environmental health are all connected.
We talked about whole genome sequencing and how it was so key to identifying this MFSD8 duplication. It's an amazing tool, isn't it?
It really is. It's totally changing how we approach genetics research. Whole genome sequencing lets us look at an individual's entire genetic code. We get this complete blueprint of their DNA.
Like a super detailed map of their genes.
Yeah, exactly. And in this study, it allowed them to find the exact spot of that MFSD8 duplication. And that led to the development of that targeted genetic test for small Swiss hounds.
Which could really make a difference. Now, breeders can identify carriers and make informed choices about breeding, hopefully preventing a lot of puppies from being born with NCL7.
It really shows how scientific discoveries can lead to real, tangible benefits for animal health. And as whole genome sequencing becomes more affordable and accessible, we're going to see its impact grow even more.
It feels like we're entering a new era in veterinary medicine.
Yeah.
Where we can use genetics to diagnose, treat, and prevent so many different diseases.
I think so too. And this study reminds us that every discovery, no matter how small it seems, can have these ripple effects that go way beyond that initial finding.
Well, I think we've done a great job of breaking down this research. We've covered the genetics, the methodology, the implications. We even talked about things like gene therapy and genetic diversity.
We've covered a lot of ground, and I hope our listeners have a better grasp of the power of canine genetics and the incredible potential it has to improve the lives of animals and humans.
This research is a great example of the dedication and creativity of scientists around the world, working hard to understand the complexities of genetics and trying to find solutions to these challenging health problems.
It's a really exciting time to be in this field. Who knows what incredible discoveries are just around the corner?
On that note, I think it's time to wrap up this deep dive.
It's been a pleasure joining you today.
And to all our listeners, thank you for being here with us on this journey. We hope you've learned something new and that you're as excited as we are about how genetics is shaping the future of veterinary medicine.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.